Right, so we're going to have a look at the first stage of protein synthesis, which is called transcription. So let's have the scenario where we are going to have a look at how on a particular chromosome at a specific locus we have got the gene that codes for the enzyme amylase. So what's going to happen is this gene is going to be used to ultimately synthesize the amylase protein. Remember, we're looking at the process in a eukaryotic cell, so transcription takes place in the nucleus, in the cytoplasm of the nucleus. So here we go. A lot of people often have got a misconception that DNA is some sort of molecule that just stores the genetic code. DNA is being used and is being accessed all the time. So if we think about our digestion topic, in the salivary glands, in those cells, amylase enzyme, salivary amylase, needs to be synthesized constantly. So the gene that codes for amylase is being used all the time. In those cells, at the ribosomes, on the rough endoplasmic reticulum, amylase enzyme is being synthesized constantly. Okay, so here we go. What happens then in transcription? Initially, then the gene, so that specific sequence of DNA bases, the double helix, first of all, has to unwind itself and then unzip. And so an enzyme that we've discussed previously, helicase, comes along and breaks the hydrogen bonds between the bases, but only at the bases within the gene. The rest of the DNA molecule remains in its double helix state. So often students confuse DNA semi-conservative replication with transcription. This is one of the big differences. The DNA bases only within the gene have their hydrogen bonds broken. If we look at that particular gene in a bit more detail, I've come up with DNA-based sequences. Obviously, it's very short. It's much shorter than it actually is, but this is just for illustrative purposes. So the first thing to say is it's only one of the DNA polynucleotide strands. Remember, a DNA molecule is made up from two DNA polynucleotide strands. Only one of those strands is going to be used, and we refer to that strand as being the template strand. Now, in this case, it's the one on the top here. In a question that you might get, it could be on the bottom, but only one of those polynucleotide strands is going to be used to form the pre-mRNA molecule. As discussed previously, we are going to read the sequence of DNA bases in threes. So the first triplet code is TAC, the second is CCC, and then we have TAC again, and then AGG. So that's our DNA base sequence on the template strand. What's going to happen then is this. Within the cytoplasm of the nucleus are free RNA nucleotides. So if I draw them like so we have free RNA nucleotides. I'm just going to represent them like so. So this just represents the phosphate and sugar, the sugar phosphate backbone of one nucleotide. We're not really interested in either of those two components, it's the base that's of significance. So here we've got a free RNA nucleotide with an adenine base. Here's one with a cytosine base. Here's one with a guanine base. And finally, one of the big significant differences, as we've looked at previously, between RNA and DNA is that in RNA, the nitrogenous base of thymine doesn't exist. It's replaced by the nitrogenous base uracil. So we've got free RNA nucleotides with bases A, U, G, C. Uracil still is able to do complementary base pairing with adenine. Simply, uracil replaces thymine in RNA. So when the gene at the locus amylase, the gene locus amylase, when the hydrogen bonds are broken between the DNA bases, we've now got our two separate DNA polynucleotide strands, and on the template strand, complementary base pairing between the exposed DNA bases and the free RNA nucleotides starts to take place. So to start off with, we would get complementary base pairing like so. So we would have adenine, and then we would have a uracil, and then we would have a guanine. You can pause the video now, and hopefully you could do complementary base pairing of the rest of the DNA bases on the template strand. If you continue, then we would get the following taking place. And you can see I've made a mistake there already, so that's going to be uracil. So that's the first step within with transcription. At the moment, what's important is that these are individual RNA nucleotides that have complementary base paired by carrying out hydrogen bonds, specific hydrogen bonds, between the exposed bases on the template strand. But they are individual nucleotides at the moment. What happens next is an enzyme will come along, and that enzyme is called RNA polymerase. And RNA polymerase will run along the length of the RNA nucleotides, and it catalyzes condensation reaction between those nucleotides. It forms a phosphodiester bond between the individual nucleotides, and so ultimately, I just run from that end, each of those nucleotides are joined together, and so now what's been formed is a pre-mRNA strand. We have a pre-mRNA molecule, and as we discussed before, RNA molecules are just made up from single polynucleotide strands. Just to illustrate the principle that we discussed previously about within a gene you find coding and non-coding sequences of bases. Let's just say, for argument's sake, and our very, very short gene that just here, this is going to be an intron, and therefore the rest of the DNA base sequence is coding exon. I'm going to use that in a moment just to illustrate a point. A couple other bits of terminology that we can look at. Whereas previously we talked about the fact that when we look at DNA, so here are our sequence of DNA bases, a sequence of 3 DNA bases is called a triplet code, as we discussed before. The corresponding sequence of 3 bases on RNA, messenger RNA, so this corresponding sequence here then of AUG, we just call the sequence of 3 bases on RNA, whether it's messenger RNA or we're going to look at tRNA later on, we just refer to that as a codon. So a codon is a sequence of 3 bases. It's the RNA equivalent of a DNA triplet code. You will have a copy of a messenger RNA codon table, and what that messenger RNA codon table does is show you the specific sequence of bases that mRNA codons are ultimately going to code for, and we can use that table a little bit later on. So at the moment, let's get back to the fact that pre-mRNA molecule has been formed, and just to remind ourselves then what happens next is the pre-mRNA molecule will detach from the template strand, and then what will happen is we have the act of splicing, and this is where the introns are removed from the pre-mRNA strand. So on our pre-mRNA strand, we have the sequence, where I went wrong earlier, we have the sequence of AUG. So that is an intron. So what will happen in splicing is that intron will be removed. So we'll be left with AUG, G, G, G. The intron is removed, and then we have U, C, C. So the intron has been removed. So we can now just refer to this as an mRNA molecule. Once it's gone through splicing, that is an mRNA molecule. And so mRNA molecules are shorter, and they'll have a lower mass as well, due to the fact that introns will have been removed. In eukaryotic cells then, next, that mRNA molecule will move to ribosomes and will be involved in the process of translation. Just in terms of the DNA molecule, once the pre-mRNA strand has moved away, that DNA molecule will then have its hydrogen bonds between the bases will reform, and the DNA molecule will just form its double helix shape again, once transcription has occurred. Just two final bits of terminology that we need to discuss, and the first is the genome. So when we talk about the genome of an organism, that's referring to the complete set of genes within all of its DNA molecules. So that's just referring to all of the genes that it has. And then another phrase is the proteome, and that's referring to all of the proteins which can be produced by that organism's genome. So genome, complete set of genes that an organism contains, and the proteome, which is all the proteins which can be produced by that genome.